The Capensis problem, a One Health concept by Group 8. These are the topics we'll be discussing.
Capensis versus clone.
The Cape honeybee, Apis mellifera capensis, is unique as its worker bees are able to lay diploid eggs that develop into female workers without requiring fertilization. This phenomenon is known as Thelytokis parthenogenesis. The offspring of these female worker bees are referred to as pseudoclones, due to their genes being virtually identical to those that laid them. African honeybees, the Apis mellifera scutellata, do not possess this characteristic, and if worker bees lay, they will produce haploid eggs that develop into drones only. How the problem arose. In 1990, commercial beekeepers moved colonies of the Cape honeybee via direct transfer of 80 colonies from the north of Cape Town to the north of Joburg, and moved approximately 400 Apis mellifera scutellata colonies from KZN to the Cape for pollination, which then returned infested with the Cape honeybee. In 1995, the Capensis problem was acknowledged, and the scientific recommendation to stop the Capensis from invading native hives was to stop the migration of bees for 2 to 3 years. Due to the annual value attribution of honeybees in commercial crop pollination being around 3.2 billion rand, commercial beekeepers did not comply, and the problem persisted.
Inside the bee.
Queens are the reproductive individuals in the colony. The queen only mates with the drones once and stores their sperm in her spermatheca for life. She then regulates the fertilization of her eggs. Those laid in worker cells are fertilized, while those laid in drone cells are not. In the absence of a queen's pheromones, some worker bees will start to lay unfertilized eggs until a new queen is reared from her offspring. Thelytoki in the Cape honeybee is shown to be under the control of a recessive gene, a gene that influences rapid ovary activation and queen pheromone synthesis, thus enables these morphologically worker bees to develop into false queens. As facultative parasites, the Capensis worker females actively seek out and invade susceptible AM scutellata colonies where they produce a queen-like pheromonal bouquet. The Capensis bee is not controlled by the host queen, and a pheromonal contest between the two ensues. This distorts the established hierarchy of the hive, results in rejection of the host queen by her own workers, and leads to her death. The queen's mandibular glands produce the QMP complex in a caste-specific manner, where stearic acid is converted into the main mandibular gland pheromone component through a bifurcated 3-step process. This complex attracts workers to attend the queen and is responsible for the social organization of the colony. In the absence of the queen, the workers employ the molecular pathways usually utilized by queens to produce 9-ODA, known as the queen substance.
Inside the hive.
When the Capensis worker bees invade, they take over the reproduction of the host colony and produce large numbers of pseudoclones or false queens. As the emerging bees are all essentially reproductive and existing drones are no longer under the influence of the host queen's pheromones, no foraging is done, and nectar and pollen reserves are depleted. These colonies diminish in size, eventually collapse, and the Cape honeybee just disappears. When the host queen is lost, Capensis false queens monopolize reproduction at the expense of her original offspring. The host queen's workers or drones neglect her eggs and larvae, and the genetic potential of that specific queen line of AM scutellata is eventually lost.
Hive to hive and hive to human.
According to the ARC, the Capensis problem causes approximately 30% loss of the African honeybee colonies per year. This is due to drifting Cape bees that actively invade colonies of AM scutellata when placed in the same apiary, hive-to-human problems that include critical losses of pollination of agricultural crops and natural vegetation, as well as a decline in honey and wax production, and economic losses of approximately 16 billion rand annually that we face. This includes almost 180,000 jobs within the honeybee industry.
Prevention is key to saving the bee.
According to the Agricultural Pests Act, all Capensis-infested colonies must be destroyed, but it is the beekeeper's responsibility to detect the infestation first. Outside the hive, one will see reduced foraging, dead bees, bees fighting at the entrance of the hive, and so-called black bees. Inside the hive, one will see worker bees fighting on the comb and workers with a typical laying behavior, with distended abdomens, shiny appearances, and a wings out posture. The ARC suggests that keeping AM scutellata in smaller colonies, harvesting the honey more often, and ensuring fewer bee migrations will be preventative.
The stakeholders.
There is a deficit of government body involvement in the bee industry. We deem it necessary for the intervention to shift the position of bees from plant health to animal health under legislative structures. There is also a shortage of communication and data recording amongst the different stakeholders that need to be addressed. Stakeholders including the beekeepers, migratory beekeepers and Aedes scutellata produce farmers, and the crop farmers, DAF, the ARC, and entomologists in educational institutions.
Heroes of the bees.
The veterinary industry has a bigger role to play in the mitigation of the Capensis problem. Dr. Yusuf and Prof. Perk, Mr. Mike Allsop, the ARC Plant Protection Research Institute and National Department of Agriculture are all major researchers of the Capensis problem. And though there are more, we feel the responsibility lies with the Department of Agriculture to implement regulations concerning the management of this problem.
Other negative impacts.
Varroa destructomites, which are also associated with chalk brood within the hive, insecticides that are used by agricultural practices, and their habitat, which is largely lost through human development.
Ow! What was that? Maybe this time.